Ready to have your mind blown.
Mm.
Today, we're diving into how bacteria pull off a cellular heist to infect us. And I know what you're thinking, ew, bacteria, but trust me, this is fascinating stuff. It has some pretty big implications for the future of medicine, too.
It really is amazing when you think about it. These tiny organisms have evolved these insanely complex ways to survive.
For real. And the mastermind we're focusing on today, Shigella, that bacteria that causes that awful intestinal infection, dysentery.
Yeah, unfortunately, it's still a big problem, especially in parts of the world without access to clean water. And kids, they're hit the hardest.
Exactly, which is why I was so interested in this new research. It's not just about figuring out Shigella, but about uncovering weaknesses in its attack plan. Weaknesses that might apply to other infections, too. So, paint us a picture here. How does Shigella actually get us sick?
Okay, so imagine this. Like any good thief, Shigella has to get inside to take what it needs to replicate, right? Our cellular resources. Only problem is our cells. They've evolved some pretty serious defenses to keep those kinds of invaders out.
So how does Shigella get past those defenses? I mean, it's not exactly like it just breaks down the door, right?
Not quite. The Science Daily article we're looking at compares it to a heist, which I love. Instead of brute force, Shigella is sneaky, like a master manipulator. It releases proteins that totally disrupt how our cells normally work, creating this perfect distraction so it can slip in unnoticed. They call it messing up the host to survive.
Okay, that's intriguing, but messing up the host seems kind of vague. What's actually going on at the, molecular level?
So picture this. Shigella releases these proteins, right? Yeah. And those proteins, they mess up the balance inside our cells. The acidity changes, things get more acidic, and suddenly the cells are making way too many lipids.
And just so everyone's on the same page, lipids are those building blocks of our cell membranes,
Yeah,
right?
exactly. And having too many lipids, well, it's like throwing a wrench in the gears. It messes everything up.
So it's like Shigella is causing all this chaos inside our cells, throwing everything out of whack. But where do these proteins, TOM1 and polyp that the article kept mentioning, come in?
Ah, those are important ones. Okay, so think of 2M1 and TOLIP like the cleanup crew for our cells.
Okay.
Their whole job is to find and trash any proteins that are just hanging around, cluttering things up. Basically, they're like the ultimate garbage collectors, right?
Yeah. Keeping everything neat and tidy, making sure the cell runs smoothly. Got it. So how does Shigella mess that up with all the chaos it's creating?
Well, usually TM1 would be busy cleaning up those extra lipids that Shigella is throwing around. But the thing is, that acidic environment and all those lipids, it basically throws TM1 totally off.
Like overwhelming the cleanup crew with way too much garbage so
Yeah.
they can't do their job.
Exactly. And this is the really sneaky part. The researchers found that when TM1 gets all confused like that, it can't bind to the lipids properly.
Hold on, what do you mean by bind?
Good question. Basically, it's like imagine two puzzle pieces fitting together perfectly.
Right, yeah.
Yeah, that's kind of what's happening, but on a molecular level. T1 has to bind to the lipids to recognize them.
Yeah.
It's something that needs to be taken out. But Shigella, it messes that whole process up.
So not only is Shigella using lipids to cause chaos, it's like it's using them to hide from our defenses too, making it harder for TM1 to find them.
Exactly. And this is where it gets really interesting from a research perspective. Because now that they understand how this binding works and how Shigella disrupts it, scientists think they might be able to, block it.
Okay, so now we're talking about stopping Shigella from hiding out and multiplying in our cells, disarming it.
Exactly. Imagine if we could stop that binding from being disrupted. TOM1 could get back to work, clean up the mess, and our cells could actually fight back.
That's amazing. So this discovery has implications beyond just Shigella then, right? The article hinted at that.
Absolutely. And this is what's so exciting. This whole binding thing that Shigella is taking advantage of, it might be a strategy that other bacteria use too.
You're kidding. So this could change how we think about, all bacterial infections.
Think about it. What if we could use these findings to fight things like pneumonia or UTIs, even staph infections? The possibilities are huge.
Man, this is what I love about science. A breakthrough in one area, like understanding how Shigella infects our gut, could lead to solutions for so many other problems. It shows how everything is connected.
For sure. But we do have to remember that this is still early days, research-wise.
Right, of course.
We've got this important piece of the puzzle, but there are still a lot of questions, like how do we actually block this binding safely? And not just in a lab, but in people.
So the next step is translating this knowledge into actual treatment.
Exactly. But the fact that we found this weakness, this Achilles heel, in Shigella's plan, that's a huge deal.
It's like we've got Shigella's playbook and now we can plan our counterattack.
Right. And here's something else to think about. The article mentioned that Shigella does all that damage in, like, the lining of the intestines,
Yeah,
right?
that's its thing, wreaking havoc in our guts.
Well, get this. The lining of our intestines, it's actually pretty similar to the lining of other organs, other systems in our bodies. So, like, could this same strategy be happening in other infections? Infections that target other parts of us?
Yeah.
Pretty wild, huh?
Now that you mention it, the article did say that this whole binding mechanism might not be unique to Shigella. So could this be like a universal thing, a way that bacteria have evolved to target us everywhere?
Imagine that. What if we could develop treatments that, like, interfere with this binding? It could have a huge impact. Pneumonia, UTIs, even wound infections. We could target them all in a whole new way.
Now that is revolutionary. It really shows how a discovery about one thing, like Shigella in the gut, could unlock these solutions for so many other health problems. It's like it all comes back to the same basic mechanisms.
Exactly. It just goes to show you the power of scientific research. Every discovery, even the small ones, can unlock these huge secrets with implications we can't even imagine yet.
So, to anyone listening who might have been thinking, who cares about some microscopic bug? I hope this deep dive has shown you how important this discovery could be. It's not just about Shigella, it's about understanding how our bodies work at the most basic level, and using that knowledge to fight off infections in a smarter, more effective way.
It's about going beyond just treating symptoms. It's about getting to the root of the problem, giving our bodies the tools they need to defend themselves, no matter what's attacking.
This kind of research really makes you think, what other tiny battles are going on inside us right now, and what other incredible discoveries are still out there just waiting to be uncovered?
That's the beauty of science, right?
Mm.
Always more to learn, more to explore. Every new finding opens up a whole world of possibilities. Possibilities we can only imagine.
And on that note, we'll wrap up this deep dive into the fascinating world of cellular hijacking. Until next time, everyone, keep those brains buzzing.